everyone, I am Shwelidash welcoming you to the Medical and Pharmacy Crash Course Division in the Knowledge Club online. In the previous episode, we learned about synapses and definition of neurotransmitter. Today, I will discuss the various types of neurotransmitter. By the end of this section, you will be able to know about various types of neurotransmitters which are bound to the neuroreceptors. Stay tuned up to end because if you skip the lecture, you can miss valuable points in between lecture for your exam. So stay tuned up to end. I am going to start today's episode. Let's start. Neurotransmitters are the substances that mediate chemical signaling between neurons. For a substance to be considered a neurotransmitter, it must meet several generally recognized criteria. First, the substance must be demonstrated to be present in the presynaptic terminal, and the cell must be able to synthesize the substance. It should be released on depolarization of the terminal. Finally, there should be specific receptors for it on the postsynaptic membrane. This last criterion is certainly true for substances that act as synaptic transmitters. But if we want to be inclusive and include substances that act over widespread territories rather than just as a single synapse, the last criterion needs to be relaxed to include situations in which receptors are located at sites outside the synapse. Neurotransmitters have been suggested as a general term to describe both synaptic and non-synaptic signaling between cells. More than 100 substances have been identified as potential neurotransmitters because they have met hence the potential equivalents or all of these criteria. These substances can be subdivided into three major categories. That is, number 1, small molecule transmitter, number 2, peptides, and number 3, the gaseous transmitters. The small molecule neurotransmitters may be further subdivided into acetylcholine, amino acid, biogenic amines, and purines. The first three groups of small molecule transmitters contain that are considered the classic neurotransmitters. Remaining transmitters are substances that are more recent addition to the list of neurotransmitters, although many of them have been known as biologically important molecules in other contexts for a long time. So, I am going to start with the small molecule neurotransmitters. Number 1 type, that is acetylcholine. In the peripheral nervous system, acetylcholine is the transmitter at the neuromuscular junctions, at sympathetic and parasympathetic ganglia, and of the postganglionic fibers from all parasympathetic ganglia and a few sympathetic ganglia. It is also a transmitter within the CNS, most prominently of neurons in some brainstem nuclei in several parts of the basal forebrain, septal nuclei and nucleus basalis, and basal ganglia, and in the spinal cord, that is motor neuron axon collaterals. Cholinergic neurons from the basal forebrain areas project diffusely throughout the neurocortex and to the hippocampus and amygdala, and they have been implicated in memory functions. Indeed, degeneration of these cells occurs in Alzheimer's disease, a form of dementia in which memory function is gradually and progressively lost. Acetylcholine is synthesized from acetyl coenzyme A and choline by the enzyme choline acetyltransferase, which is located in the cytoplasm of cholinergic presynaptic terminus. After synthesis, acetylcholine is concentrated in vesicles. After release, the action of acetylcholine is terminated by the enzyme acetylcholinesterase, which is highly concentrated in the synaptic cleft. Acetylcholinesterase hydrolyzes acetylcholine into acetate and choline. The choline is then taken up by an sodium ion syn-porter in the presynaptic membrane for the resynthesis of acetylcholine. The extracellular enzymatic degradation of acetylcholine is usual for a neurotransmitter in as much as the sympathetic action of other classic neurotransmitters is terminated via reuptake by a series of specialized transporter proteins. Second type of small molecular neurotransmitters are the amino acids. A variety of amino acids function as neurotransmitters. The three most important are glutamate, glycine and GABA, GABA. Glutamate is the neurotransmitter at the overwhelming majority of excitatory synapses throughout the CNS. Despite its ubiquity, it was initially difficult to identify specific neurons as glutaminergic because glutamate is present in all cells. It has a key role in multiple metabolic pathways and it is precursor to GABA, the major inhibitory neurotransmitters. Nevertheless, experimental results have now clearly established glutamate as the major excitatory CNS neurotransmitter. When applied to cells, it causes depolarization. It released from neurons and specific receptors and transporters for it have been identified. In addition to being the main excitatory neurotransmitter, glutamate is a potent neurotoxin at high concentrations. Thus, strict limitation of glutamate's activity after its release from the presynaptic terminal is necessary, not only to allow normal synaptic transmission but also to prevent cell death. This task is accomplished by specialized membrane transporter proteins. GABA and glycine act as inhibitory neurotransmitters. GABA is the major inhibitory transmitter throughout the nervous system. GABA is produced from glutamate by a specific enzyme, the glutamic acid decarboxylase. Glutamic acid decarboxylase enzyme that is present only in neurons that use GABA as a transmitter. Thus, experimentally, it is possible to identify cells as inhibitory GABAergic neurons by using antibodies to this enzyme to mark them. Many local interneurons are GABAergic. In addition, several brain regions contain large number of GABAergic potential neurons. The most notable are the spiny neurons of the striatum and the Purkinje cells of the cerebellar cortex. The inhibitory nature of Purkinje cells are especially surprising because they represent the entire output of the cerebellar cortex. And thus, cerebellar cortical activity basically functions to suppress the activity of the downstream targets, the cerebellar and vestibular nuclei. Glycine. Glycine functions as an inhibitory neurotransmitter in a much more restricted territory. Glycinergic synapses are predominantly found in the spinal cord, where they represent approximately half of the inhibitory synapses. They are likewise present in the lower brainstem, cerebellum and retina in significant numbers. Interestingly, glycine also has another synaptic function as excitatory NMDA type glutamate receptors. Glycine must also be found for the ion channel to open. Thus, it acts as a co-transmitter as these synapses. It was generally thought that under physiological conditions, the extracellular glycine concentrations was high enough that the glycine binding sites of the NMDA channel were always saturated. But recent results suggest that this may not always be true, which implies that Fluctuations in glycine levels may also be an important modulator of NMDA-mediated sympathetic transmission. After GABA and glycine are released from the presynaptic terminus, they are taken back into the nerve terminal and neighboring glia by high-affinity sodium ion and chloride ion coupled membrane transporters. These sodium ion and chloride transporters are part of which is called the solute carrier 6, means the SLC6 family of transporters that also includes those for the biogenic amine neurotransmitters. But it is distinct from those for glutamate. Transport of the neurotransmitter into the cell is accomplished by syn-port with 2 sodium ion and 1 chloride ion. 4 GABA transporters GAT1, GAT2, GAT3 and BGT1 genes have been identified till now. However, GAT1 and GAT3 are the ones that are highly expressed in the CNS. Depending on region and species, they may be expressed in neurons and or in glia. There are 2 main glycine transporters, GlyT1 and GlyT2. GlyT1 is found predominantly on astrocytes and is present throughout the CNS. In contrast, GlyT2 is located on glycinergic nerve terminals and is largely restricted to the spinal cord, brainstem and cerebellum. At least 5 transporters EAAT1, EAAT5, in which EAA stands for acetylcholine amino acid transmitters that carry glutamate across the plasma membrane have been identified. They are all part of the sodium ion potassium ion dependent family of transporters. Inward movement of each glutamate molecule is driven by the co-transport of 3 sodium ions and 1 hydrogen ion and the counter-transport of 1 potassium ion out of the cell. In addition, the transporter has chloride conductance. Although passage of chloride ions is not stoichiometrically linked to glutamate transport. Glutamate transporters are found on both neurons and glia. However, the transporters differ in their regional and cellular distribution and in their pharmacological and biophysical properties. For example, EAT2 is found on glia and is generally responsible for more than 90% of glutamate uptake from the extracellular space. The glutamate uptake into glial cells by EAAT2 is eventually returned to the presynaptic terminal by the glutamine cycle inside glial cells. Glutamate is converted to glutamine. Glutamine is then transported out of the glial cell and back into the presynaptic terminal, where it is subsequently converted back to glutamate. Glutamate inside the presynaptic terminal is packaged into synaptic vesicles by the second set of glutamate transporters known as VGLUT is the vesicular glutamate transporters which are present in the membrane of glutaminergic vesicles. Transport of glutamate into synaptic vesicles by VGLUT is driven by the counter transport of hydrogen ions, the electrochemical gradient for which having been established by an hydrogen ion ATPase in the vesicle membrane. Third neurotransmitter is the biogenic amines. Many of the neurotransmitters in this category may be familiar because they have role outside the nervous system, often as hormones. Among the amines known as act as neurotransmitters are dopamine, norepinephrine or noradrenaline, epinephrine or adrenaline, serotonin, 5-hydroxytryptamine or means 5-HT and histamine. Dopamine, norepinephrine and epinephrine are catecholamines and they share a common biosynthetic pathway that starts with the amino acid tyrosine. Tyrosine is converted to L-dopa by the enzyme tyrosine hydroxylase. L-dopa is then converted to dopamine by dopa decarboxylase. In dopaminergic neurons, the pathway stops here. In noradrenergic neurons, another enzyme, dopamine beta hydroxylase, converts dopamine to norepinephrine. Epinephrine is obtained by adding a methyl group to norepinephrine via phenylethylamine N-methyl transferase. In serotonergic neurons, serotonin is synthesized from the essential amino acid tryptophan. Tryptophan is first converted to 5-hydroxytryptophan by tryptophan 5-hydroxylase, which is then converted to serotonin by aromatic L-amino acid decarboxylase. Finally, in histaminergic neurons, the conversion of histidine to histidine is catalyzed by histidine decarboxylase. Removal of sympathetic synaptically released biogenic amines is generally accomplished by reuptake into glia and neurons via transporters belonging to the sodium and chloride dependent transporters family. The catecholamines are often degraded by two enzymes, monoamine oxidase and catechol-O-methyl transferase. Within the CNS, nerve cells that use biogenic amines as neurotransmitters are primarily found within one of a few brainstem nuclei, most of which project rather diffusely throughout large areas of the brain. Noradrenergic neurons are primarily found in the locus coeruleus and nucleus subcoeruleus, which are located near each other in the dorsal part of the rostral bones. The neurons of the locus coeruleus project throughout the entire brain. Targets of the nucleus subcoeruleus are more limited but still widespread and include the pons, medulla and spinal cord. Norepinephrine is also important in the peripheral nervous system because it is used by postganglionic sympathetic cells. Serotonergic fibers arise from a series of nuclei located at the midline of the brainstem, known as the Raphe nuclei. Similar to the noradrenergic fibers, serotonergic fibers are distributed throughout the most of the brain and spinal cord. Dopaminergic fibers arise from two main brainstem regions, the substantia nigra pars compacta, which projects to the striatum, and the ventral tegmental area, which projects more widely to the neocortex and subcortical areas, including the nucleus accumbens. Histaminergic neurons are located within the tuberomammillary nucleus of the hypothalamus and project diffusely throughout the CNS. Finally, adrenergic neurons are relatively few in number when compared with the other biogenic amine transmitters. They have cell bodies localized to small cell groups in the rostral medulla. The largest group, termed C1, has projections to the locus coeruleus and down to the thoracic and lumbar levels of the spinal cord, where they terminate in the autonomic nuclei of the intermediolateral and intermediomedial cell columns. Thus, these neurons are important for autonomic functions, particularly vasomotor ones such as control of arterial pressure. The diffuse nature of the projection pattern of most of the amine system is minored in their proposed functions. Activity in the defined aminergic system is believed to be important in setting global brain states. For example, these systems are involved in setting the level of arousal, that is sleep, walking, attention and mood. Their involvement in pathways connected with the hypothalamus and other autonomic centers also indicated that they have important homeostatic functions. The role of dopamine is balancing the flow of activity through the basal ganglia pathway and how it heads to the motor symptoms observed in Parkinson's disease. Fourth type of neurotransmitters are purines. ATP has the potential to act as a transmitter or co-transmitter at the synapses in the peripheral and central nervous system. ATP is found in all synaptic vesicles and thus is co-released during synaptic transmission. ATP has its own receptor which, like standard neurotransmitters, are coupled to ion channels, but it can also modify the action of other neurotransmitters which it is co-released, including norepinephrine, serotonin, glutamine, dopamine, and GABA. Glial cells may also release ATP after certain types of stimulation. Once released, ATP is broken down by ATPase and 5 nucleotidases to adenosine, which can be taken up again by the presynaptic terminals. Fifth neurotransmitters is the peptides. Peptide neurotransmitters consist of chains of between 3 and about 40 amino acids. Studies of neuropeptides focused on the hypothalamus for many years. However, it is now clear that neuropeptides are released by neurons and act on receptors throughout the CNS, and thus are a fundamental mechanism of neurotransmitter throughout the CNS. To date, more than 100 neuropeptides have been identified. They can be classified into several functional groups which list some of the known neuropeptides. It is now clear that many neurons that release classic neurotransmitters also release neuropeptides. I will discuss later the understanding the interaction between coexisting classic and peptide neurotransmitters has become an important area of research. In addition to being co-released with another transmitters, neuropeptides can also function as the solo or primary neurotransmitter as a synapse. In some ways, neuropeptides are like the classic neurotransmitters. They are packaged into synaptic vesicles. Their release is dependent on the calcium ion and they bind to specific receptors on target neurons. However, there are also significant differences that have led to alternative names for the intercellular communication mediated by neuropeptides, such as non-sympathetic, parasympathetic, and volume transmission. Unlike classic neurotransmitters, which are synthesized at the presynaptic terminal, neuropeptides are synthesized at the cell body and then transported to the terminal. Neuropeptides are packaged into large electron-dense vesicles that are scattered throughout the presynaptic terminal rather than in small electron-lucent vesicles locked at the active zone, where small molecule transmitters are stored. In neurons that map multiple neuropeptides, that various peptides are co-stored in the same vesicles. Neuropeptide receptors are not confined to the synaptic region, and in general, peptide action is not limited by reuptake mechanisms. Each of these differences has functional implications. For example, the separate storage of peptide and non-peptide transmitters immediately raises the question of whether the two transmitters are co-released or differentially released in response to particular stimulation patterns. In fact, differential release of peptide and classic transmitters from the same cell has been demonstrated for several types of neurons and is probably a result of the differences in vesicle storage described in earlier episode. Because of their proximity to the active zones, non-peptide vesicles can be released rapidly, less than 1 millisecond, in response to single action potentials as a result of localized influx of calcium ion. Thus, low frequency stimulation of the cell causes just the release of non-peptide transmitter. In contrast, with higher frequency stimulation of the presynaptic neuron, there is a more global increase in calcium ion throughout the nerve terminal that leads to release of neuropeptide as well as neurotransmitter. When neuropeptides are co-released with other transmitters, they may act synergistically or antagonistically. For example, in the spinal cord, tachykinin and calcitonin gene related peptide, the CGRP, act synergistically with glutamate and with substance P to enhance the action of serotonin. Conversely, tachykinins and CGRP antagonize norepinephrine's action at other synapses. In interactions, however, are not simply a one-to-one synergism and antagonism at a particular synapse because of the differing temporal and spatial profiles of the action of peptides versus classic transmitters. In particular, the slower release and lack of rapid reuptake mean that neuropeptides can act for long durations, diffuse over a region of brain tissue, and affect all cells in that region that have the appropriate receptors rather than just acting at the specific synapse at which it was released. In fact, studies have shown that there is often a spatial mismatch between the presynaptic terminus that contain a particular neuropeptide and the size of the receptors for that peptide. In some, peptides released from a particular synapse probably affect the local neuronal populations as a whole, whereas the co-released classic transmitters act in more of a point-to-point manner. Sixth neurotransmitter is the opioid peptides. Opioids are drugs derived from the juice of the opium poppy. Compounds that are not derived from the opium poppy but that exert direct effects by binding to opioid receptors are called opioids and form a clinically and functionally important class of neuropeptides. Operationally, opioids are defined as compounds whose effects are stereospecifically antagonized by a morphine derivative called naloxone. The three major classes of endogenous opioid peptides in mammals are enkephalin, endorphin, and dynorphin. Enkephalins are the simplest opioids. They are pentapeptides. Dynorphins and endorphin are somewhat longer peptides that contain one or the other of the enkephalin sequence at their N-terminal ends. Opioid peptides are widely distributed in neurons of the CNS and intrinsic neurons of the gastrointestinal tract. The endorphin are discretely localized in particular structures of the CNS, whereas the enkephalin and dynorphin are more widely distributed. Opioids inhibit neurons in the brain involved in the perception of the pain. Indeed, opioid peptides are among the most potent analgesic, pain-relieving compounds known, and opioids are also therapeutically as powerful analgesics. They exert their analgesic effect by binding to specific opiate receptors. Seventh neurotransmitter, that is substance P. Substance P is a peptide consisting of 11 amino acids. It is present in specific neurons in the brain, in primary sensory neurons, and in plexus neurons in the wall of the gastrointestinal tract. The wall of the gastrointestinal tract is richly innervated with neurons that form networks or plexuses. The intrinsic plexuses of the gastrointestinal tract exert primarily control over its motor and sensory activities. These enteric neurons contain many of the neuropeptides, including substance P, that are found in the brain and spinal column. Substance P is involved in pain transmission and has a powerful effect on smooth muscle. Substance P is probably the transmitter used in synapses made by primary sensory neurons. Their cell bodies are in the dorsal root ganglia, with spinal interneurons in the dorsal horn or the spinal column, and thus it is an example of the peptide acting as a primary transmitter at the synapse. Enkephalins act to describe the release of substance P at these synapses and thereby inhibit the pathway for pain sensation at the first synapses in the pathway. Last type of neurotransmitter is the gas neurotransmitters. Neurotransmitters are highly permanent and simply diffuse from synaptic terminals to neighboring cells after synthesis. Their synthesis being triggered by depolarization of nerve terminal, the influx of calcium and activates synthetic enzymes. Moreover, there are no specific reuptake mechanisms nor do they undergo synaptic enzymatic destruction. So, their action appears to be ended by diffusion or binding to superoxide anions or various scavenger proteins. Both nitric oxide and carbon monoxide are examples of gaseous neurotransmitter. NO means nitric oxide is a transmitter at synapses between inhibitory motor neurons of the enteric nervous system and gastrointestinal smooth muscle cell. NO also functions as a neurotransmitter in the CNS. The enzyme NO synthase catalyzes the production of NO as a product of the oxidation of arginine to citrulline. This enzyme is stimulated by an increase in cytosolic calcium ion. In addition to serving as a neurotransmitter, NO functions as a cellular signal transduction molecule both in neurons and in non-neuronal cells, that is vascular smooth muscle. One way that NO functions as a signal transduction molecule are by regulating guanylyl cyclase, the enzyme that produces cGMP from GTP. NO binds to a hem group in soluble guanylyl cyclase and potently stimulates the enzyme. Stimulation of this enzyme leads to an elevation in cGMP in the target cell. The cGMP can then influence multiple cellular processes. Revise it thoroughly. I am very sure you will require less time to remember all these structures and functions. Write your opinion to me at the comment box how much you enjoyed and time take to remember the nervous tissue anatomy and physiology after viewing today's lecture. Next episode, we will discuss various types of neurotransmitter receptors. If you like this video, please like, share and comment freely. Don't forget to subscribe our channel. If you already subscribed, then thanks a lot. After subscribing our channel, please click on the bell button for getting the regular updates. I am coming with another video very shortly. Till then, goodbye.